The Smithsonian National Museum of Natural History holds over 148 million specimens, and nearly all of them are hidden away in secure collections.
Something that people really like to see are shark jaws and all the shark teeth.
We should be able to open any of these drawers and blow your mind.
Nearly everything in this archive is available for scientific research to solve real-world problems, like helping planes avoid birds and protecting the country from invasive murder hornets.
Their head is very robust, bright orange. That's pretty distinctive.
Now, the Smithsonian is racing to build a digital Noah's Ark.
I do think that a lot of the species that we have represented here are going to be extinct, and collections like this are going to be the only place where you can find any evidence of their existence.
It's a DNA library of all complex life on Earth, supercharged with high-resolution cameras, AI, and robots.
And so what used to take us a month, we can now do in less than a day.
This data could unlock new medicines, fuels, and ways to grow food. So how is the Smithsonian future-proofing its collections? And what mysteries hide behind locked drawers?
We can't talk about this. Oh, I could tell you, but then I would have to kill you, so.
You can walk through all of the exhibits in the National Museum of Natural History in about 2 hours. But it would take days to tour the hidden collections that make up the rest of the space. More than 1.4 million square feet spread across the museum and the off-site support center. All the facilities combined are about the same size as the U.S. Capitol building.
Museum collections are incredibly important for understanding what was there, what has changed, and what might be valuable to restore.
The number of objects in the museum has grown exponentially since it was established in Washington, D.C. in 1910. New specimens arrive here in 3 ways. First, through donations, like this 8,000-pound crystal from Arkansas. Second, expeditions. That's when scientists go into the field and bring samples back to Washington, D.C. You may recognize the name of the explorer who bagged this one.
This label of harpy eagle was collected in 1902, and if you turn it over, it says, presented to Biological Survey by President Roosevelt. Can you see that? That'd be Teddy.
Third, the museum can purchase specimens. However, this is rare. That's because the museum doesn't have a budget for it. The funds have to come from a private backer. In recent years, that's made it hard to compete with wealthy collectors for things like fossils.
After the great success of the Jurassic Park movies, every billionaire and tech bro in the country wanted to have their own private dinosaur.
The process to preserve and store a new specimen varies depending on what it is. Some animals get put on ice. Others stay fresh in alcohol. The museum's wet collection is filled with tiny jars and enormous vats, holding creatures like this stonefish.
Most venomous in the world. Whoops.
Fossils have to be painstakingly removed from layers of ancient rock, called matrix.
This is a carbide needle that a very dear friend of mine carved, so this is sort of like a special needle that nobody else is allowed to touch.
Specialists remove the muscles, oil glands, and other soft tissues from the birds.
We want to remove as much of the muscle that we can. We're trying to make a specimen that lasts forever and is strong. So I'm not a tailor or a seamstress, but I can sew up a bird pretty good.
At the museum support center just outside Washington, D.C., any recently living creatures are cleaned with the help of a non-human workforce. Carnivorous beetles.
They can get in very small nooks and crannies that we otherwise can't reach easily. They're kind of our unsung heroes.
You can't have rotting flesh in storage.
Good job, good job, good job. Thanks, guys, for all of your good work.
Clean specimens are tagged and filed away in uniform lockers. And over the past decade, the museum has also turned millions of items into digital files.
We're about to 3D scan this cactus. Absolutely do hear this in my sleep on a regular basis. If it weren't for the flash, I would be falling asleep at my desk right here, but thankfully it's there to keep me awake.
There are no giant bones, but we have actually 3D scanned other bones as well. We did a skull of the dire wolf.
Sylvia Orley helps lead the digitization effort.
I think, wow, how great is it that I get to handle these specimens every day? I just feel very blessed to have this job.
In 2022, Sylvia's team completed a 7-year effort to digitize the entire U.S. herbarium, a collection of over 5 million plants and flowers. Now they're scaling up to digitize the entire entomology collection, which is 7 times larger. A new scanner, like the setup in this video, will take up this entire room and photograph 200,000 pollinating species a year.
That's going to simplify the process greatly, where we're going to be able to digitize between 1,000 to 2,000 specimens per day using this conveyor belt.
Sylvia says this project will take about a decade. But it might be faster with AI tools, which the team is already using to reposition cameras and transcribe labels.
And I expect in the next few years, there is going to be an explosion of use so that we may be using AI for 50% of our work. But it is just in its infancy right now, so we're excited to see what's going to happen.
But not everything arrives at the Smithsonian intact. And for at least one department, that's entirely the point. In 2023, the US had over 19,000 wildlife strikes on aircraft. Almost all of those were birds. Globally, they cost the airlines billions of dollars. The Smithsonian's Feather Identification Lab is here to do something about that. It's run by Carla Dove. Yeah, that's her real name.
I get that all the time. My name is very appropriate.
Every year, her department receives up to 12,000 samples of bird remains scraped off aircraft. It's lovingly called snarge. That comes from US military slang, combining the words snot and garbage. It's up to Carla's team to use the snarge to ID the birds, so airports can use the most effective techniques to avoid them.
I asked my 4-year-old, what do I do at work? And she says, you look at dead animals. Yeah, that's true. That's what I do at work.
Sarah Luttrell is a research scientist in the bird department.
So the start to our day every day is to open these up. We get a little bit of bird. We never know how much. And then we get the data.
Over 70% of bird strikes occur below 500 feet, mostly during landing. Most of these collisions are non-fatal for humans, but if a bird gets sucked into a jet engine, the results could be catastrophic. Remember the plane that crash-landed in the Hudson River?
Less than a minute into the flight, the pilot reported a double bird strike.
In that case, we got 69 bags. Because as they went through and did the investigation, they went all the way into the engine and they wanted to know how far into the engine the feathers went.
Identifying the species of bird helps airports develop wildlife control programs and sensors to avoid collisions. If they can't physically ID a sample right away, the lab can compare its feathers to one of the 500,000 specimens in the museum's collection.
You still need the physical sample and you still need the physical expertise to do these complex identifications.
The team can get a pretty solid ID this way, but they often need extra help.
We can't be 100% sure that this feather came from a Eurasian collared dove and not a pigeon, so we're going to have to send this to the DNA lab.
The Smithsonian's Laboratories of Analytical Biology, or L.A.B. for short, is the nerve center for all things DNA.
Our big goal here is to document life on the planet. And in order to do that, the tool that we're using for biology is now genetics.
Lee White is in charge of this multi-million-dollar genome sequencing operation. A genome is the complete set of genetic material in an organism. Think of it like a blueprint for building and running a living thing. In 2009, Lee's lab helped Carla Dove's team map the genome of every known species of North American bird. That reference database has helped the Feather ID lab work more efficiently. It turns out you only need a few birds to decode the instruction manual of an entire species. Relative to other organisms, birds have a smaller genome.
We only need to do about 5 blue jays from across their range to capture all of the genetic diversity that's in blue jays. So we might have 1,000, but we only need to do 5.
Now Lee's team is working their way through the entire Smithsonian collection.
Every plate you see holds 100 DNA samples from many different projects and many different groups of organisms.
And thanks to new lab equipment, chipping away at this massive task has never been faster. This robot automatically prepares samples for DNA sequencing, a process that used to be done manually.
So this robot's really nice. I can run up to 96 samples at a time. Today we're just doing 8 because that's all I had to work with.
Katie Murphy is a genomics specialist who runs all of the instruments in the lab.
So it'll take about an hour to get through that step, but that's an hour of the robot working that I don't need to be here for normally, although it's really fun to watch it. And one of the big projects going on is doing that for all US fishes. And so right now I'm prepping samples for 8 fishes.
What used to take us a month, we can now do in less than a day. But what we end up with is magnitudes more data.
All that data is helping to build a digital Noah's Ark. It's called the Earth BioGenome Project, a global effort to finish mapping the genome of every species on Earth within the next decade. Once complete, it'll not only help scientists better understand life on a changing planet, but it could also unlock huge benefits for human civilization. This biological big swing stands on the shoulders of the Human Genome Project. A little over 20 years ago, scientists successfully mapped all 3.2 billion pairs of human DNA. In doing so, they sparked a biotech revolution. It gave scientists the tools to map, analyze, and compare genetic information at scale, transforming things like forensics, drug development, and ancestry testing. Biologists expect the Earth BioGenome Project to have similar results. But scientists estimate that 80 to 90% of Earth's species have yet to be discovered. And DNA mapping has revealed that some of the museum's own specimens, once thought to be related, are actually genetically distinct.
As we go into our collections and sequence them, we find undescribed species hidden in our collections. So the goalpost keeps moving. And so we have a long way to go.
Take these little guys.
As of right now, these 2 fish are considered to be the same species. And they look pretty similar, right? The one in my upper hand is from the Northern Caribbean. The one in my lower hand is from the Southern Caribbean. And because we've sequenced both of them, matter of fact, if I flip them over, you can see the tissue sample that we've taken from both of them. And then we ended up sequencing them. They turned out to be wildly different. And I can't tell you right now exactly why they're different anatomically, but that is exactly what I'm going to do tomorrow, is CT scan these 2 specimens so that I can learn a bit about their anatomy and be able to tell you why they're 2 different species.
So even if L.A.B. makes it through the Smithsonian's entire collection, it's just the tip of the iceberg.
Scientifically, we might have done the DNA on hundreds of thousands of species, but there's millions out there. But that's the ultimate goal, is to do it all.
It turns out that analyzing samples they do have helps Smithsonian researchers figure out where to look next. And to do that, you don't need a critter or even a piece of one. All you need is a sample of dirt or water and a kitchen blender. Chris Meyer has studied sea life in some of the world's most remote islands.
No, I don't spend a lot of time behind this desk.
He worked with Lee to develop one of ocean researchers' most important tools, the Autonomous Reef Monitoring Structure, or ARMS.
I'm a global arms dealer. Yes, we sell arms all over the world to our partners. That system was developed to mimic the nooks and crannies you find in coral reefs.
Essentially, it's just a stack of plates, but tweaking its design brought out Lee's inner MacGyver.
Home Depot, hardware store, online, science supply. It's really skunkworks fabrication stuff to help our scientists in the field process the biodiversity. So some of that is inventing things because they don't exist. We're doing things nobody else is doing.
Researchers strategically drop arms to the seafloor. Over time, plants and animals move in. After the crates are pulled back up, scientists meticulously remove, catalog, and photograph everything larger than 2 millimeters. Then they analyze the plates. All that sea life leaves behind a dazzling array of colorful patterns.
When you go back and get it after a year, each plate might look something like that. So they go from being absolutely nothing to fully populated.
We carefully take each plate apart, photograph each plate in a photo mosaic with a 3x5 grid, and we stitch those together to get massively high resolution pictures because I think those will leave a lasting legacy. It's remarkably high resolution, and that helps for the next step, which allows us to use machine learning to train, to classify each of these organisms into their functional categories.
By now, some of the algorithms can tell which species lived in the arms just by the patterns they've left behind.
Once all that's done, we scrape all the plates with paint scrapers, and we put it in a blender and grind it all up, homogenize it, make a slurry, a Reef's milkshake, and it works amazingly well. I'm pretty much a blender connoisseur, so.
I think this will be the tastiest Reef yet. Look at that, it's frothing.
I go to the different stores where you can buy blenders in the world, and the blender is the key tool.
Living things can leave behind slime, feces, or even tiny cells wherever they go. The genetic fragments trapped inside this slushy mix are enough to create a clear snapshot of the ecosystem. It's called environmental DNA, or eDNA, and it's incredibly valuable.
So we get a better sense of which are the winners and losers in these different systems, and what are the best sentinels of those change.
eDNA can also help prevent huge economic losses.
The other big utility of the arms right now is as early detectors for invasive species. Invasive species cost industries and fouling communities literally billions of dollars.
When we visited Andrea Quattrini, a research scientist, she had just used eDNA to discover a new species of soft coral that was less than a centimeter tall.
We basically just looked at the DNA in the water, and that was a sign that we have a new species that wasn't known to be there before.
That's always super exciting for biologists like Andrea.
So if we do collect a new species, I do get to describe it. You really can't describe it after yourself, but you could find probably a colleague and say, hey, I think I collected something new, why don't you describe it, and you can name it after me. I mean, we don't do that. That's like a, what's the word? Not a, what's the word? Taboo. Taboo. Yeah, it's a taboo.
Instead, Andrea named this new species after her mentor.
Parasphera sclera. I know, it's a big name. Anyways.
eDNA collected from arms is already being used to monitor ecosystems in over 3,000 locations all over the world. But biodiversity isn't just something to study. In many cases, it's something scientists are racing to save. Because over the past few decades, there's been a rapid decline in global biodiversity.
It's just inevitable that some of these insect species are going to go extinct, and collections like this are going to be the only place where you can find any evidence of their existence.
Tools like eDNA can help scientists detect species before they disappear. Sometimes, just in time to learn something from them. And that's critical, because extinction is forever. That's something Rebecca Johnson knows intimately. Today, she oversees research at the museum, but has a special place in her heart for a species on the brink.
I feel the same as everyone does about koalas. I think they're extraordinary creatures. Many people like to work on koalas because they're so iconic. They're biologically very interesting, but they're also really in need of science because there's fairly major conservation concerns for them as a species.
But the Smithsonian collection can be an irreplaceable tool for navigating an uncertain future.
Not everyone has what we have in our collections. In some cases, we might have the only examples of those specimens in the world.
And scientists are already putting them to work, not just for species conservation, but to protect the systems we all rely on.
If you enjoy food, like we all do, you might not know that we have an insect collection that's used every single day to identify potential pests that are coming into the country that may be a risk to agriculture. Similarly, the botany collection is used in the same way. We have a very huge agriculture industry here that this collection directly benefits every single day.
When it comes to defending America's farmland from invasive bugs, Matt Buffington is one of our first lines of defense.
A cicada will be like, making all this noise, and then as soon as the cicada killer grabs them and stings them, you hear the kind of it's a little sad, but that's nature.
In 2019, Matt received an urgent phone call. A homeowner in Washington state had discovered a freakishly large hornet in their backyard, and scientists needed to verify what it was as soon as possible.
And when that specimen was on its way here, I had a number of emails and phone calls asking to contact them as soon as it arrived. I mounted it. That's one part of the process, so I could examine it under the microscope. And then I went to the collection, and that's when it dawned on me. The significance of this is this might be the first record of this species in North America, and this species is detrimental to honeybee. And honeybee is a major part of our pollination system for agriculture. Vespa mandarinia, otherwise known as northern giant hornet, sometimes referred to as the murder hornet.
This invasive species from Asia had the potential to create a $100 million disaster.
Once that comes in and it gets established, you're not getting rid of it.
After Matt's positive ID, entomologists formed a SWAT team, suited up in special sting protection suits, and went on the hunt for murder hornets. Then, a single hornet tagged with a radio transmitter led them to Nest Zero, America's first established colony of murder hornets.
A few years ago, this was translocated to Smithsonian for an exhibit on invasive species to North America. And it was just in time because of the 202 individuals in the nest, 96 of them were queens, each one capable of establishing a new colony. So they got them at just the right time.
Nearly 5 years on, and there hasn't been any new sightings of the murder hornet. But these aren't the only things the collections are used for. Fossils can show us how life has adapted to past climate shifts, and unsequenced genomes might hold the key to drought-resistant crops, or life-saving medicines we haven't discovered yet.
If this collection went away, we would lose the knowledge that is the foundation of what we know about biodiversity on the planet.
Preserving that foundation isn't a given. Scientists and institutions like the Smithsonian are facing funding cuts and political pressure. Behind the headlines, that meant real uncertainty for researchers and staff, and long-term risk to one of the world's most important scientific resources. Meanwhile, private biotech companies sometimes promise they can reverse extinction with pipettes and computers. But as any ecologist will tell you, once a species is gone, it's gone. And no amount of synthetic biology can replace the complexity of real ecosystems. The clock is ticking, but if the federal government continues to invest in science and preserve what we have now, we might still shape a different future.
I think the urgency is that the technology is so good now. You can get an excellent quality genome from an old specimen, the kind of specimens that we do tend to have in this collection, and the questions are really urgent. And so not only do we have the technology to generate the data, we have the need to answer those questions with that data.
So what about the internet rumors about those giant humanoid skeletons?
We have a very large file of people asking that question.
But when we cornered the museum's top director, he set the record straight.
And where are the giant bones?
They're sloth footprints. People thought they were human footprints. And that started the whole Sasquatch giant thing. We can't get rid of it.